Really a fantastic morning. It's really just to congratulate to all of people involved to bring this community together, because I've been working on many genes in my 30 years in this field, but PCDH19 is really the most exciting. But I have to say it's also a disorder where we have seen really the biggest progress, despite perhaps our wish were not as fast as we would like to. But certainly in comparison to the other epilepsy and intellectual disability genes I've been involved with, it's really happening. So we have been involved in many different aspects on looking at PCDH19 function. But for today, I really decided to talk very specifically about sort of our work, which builds on what we published and what was mentioned very quickly this morning, and which focuses on trying to find a way how we can determine which PCDH19 variants are actually involved in the disorder and which are not. This is becoming increasingly important in human genetics, especially with the technologies now infiltrating our everyday life. Everybody's being sequenced, everybody has variants. But we really don't have the tools to interpret what these variants mean. And we heard it this morning, sometimes there's one, sometimes two, sometimes three. We find patients with 4 variants. So how are we going to really separate the wheat from the chaff in terms of importance, predictive value, and diagnosis, and of course treatment. So that's what I will try to focus on today. So in this case, the wheat is a smiley face. The chaff is not, but yeah, that can be different. I do work now with mariners, trying to help them better understand the action of the ganaxolon, so that's my disclosure for this purpose. So I start with this slide, which really shows that we came to these families, we came to these genes through studying large families, where the penetrance was really not very high, or the clinical expressivity. And we were biased by that. And when Christelle Defienne and her team discovered the same mutation in very early onset, a lot more severe phenotype, it was somewhat puzzling. But the bottom line from what you heard today from Nicola is that this disorder really have variable penetrance, not just between males and females, but among the girls with this variation. And that really complicates the analysis of everything what we do, because we really don't know why we see such variability in clinical expressivity between mom and daughter, between even monozygotic twins, as we have the example from Japan, that they are discordant in their phenotype. The best explanation probably might be in the exon activation. So this is really what we try to do. We have the black box of individual genome and individual life. We put into it even identical PCDH19 mutation, and we really end up with the spectrum of different outcomes. And these are weighted differently in different individuals, from really quite complex to almost non-penetrant. And I just have to say, if I was going to choose one major modifier, I would say that's what Paul showed on one of his last slides, that's the variability of exon activation in the brain of these individuals. We will probably never going to be able to assess it exactly in the girls, but using the mouse models, I think we at least can see how discordant can be between blood, where we really process the exon activation across many cells, and in the brain, where we can see the exon activation at the single cell level. And that's what we need to take into account. So it's to really tell you how we got into this. We were really very opportunistic back when we discovered that the gene is expressed in skin. So we have done very unbiased expression screening of these cells. At that time, Stanley was involved, and we discovered that the neurosteroidogenic system has been somehow implicated. It took us a while. We were focusing on glutamate receptors, we were looking at transcription factors, but at the end, there were really these neurosteroid metabolizing enzymes which caught our attention. And you heard also from Marina and Nicola that this allopregnanolone deficiency has been now confirmed, which was really great to see, because it took us really good 3 to 4 years to convince ourselves that actually we were seeing something real, because we measured it one way, second way. So we used 3 different methods across time to be confident that we measured it correctly. Now, there are many different ways how these enzymes and metabolites these enzymes are involved in, and that's estradiol, dehydroxytestosterone, allopregnanolone. And it's interesting, it was really good to see from Marina that many of these neurosteroids are actually deregulated. So probably that's telling us that the problem is happening a little bit earlier, a little bit more central. So this is what we found. I put it in one graph, and that been now validated, so it's very good to see, but it certainly doesn't resolve a lot, but perhaps it pushes us a little bit more to think about it, what it really means and how we can use it to help these girls. So what we also noticed that when we looked at the very interesting pattern of expression of these genes across what we studied, 12 different girls, or skin cells from 12 different girls, that we found estrogen receptor pathways being involved. Now, not only estrogen, but to some extent also androgen and progesterone. So these are really steroid hormones which are mostly known for their action in different areas. I mean, different cancers, mood, and other disorders, late onset disorders for women. They are blamed for it, or absence of some of these hormones. They are not that well really connected overall or studied in neuronal development. But when we looked, the vast majority of really significantly deregulated genes were regulated by estrogen receptor. And when you look at the pathway, pretty much all of these genes, including PCDH19, PCDH19 itself is regulated, its expression is regulated by estrogen receptor, as well as progesterone receptor. And we know that these receptors regulate each other as well, so that's why probably is quite complex. And also we started to study it, even though it didn't really make sense, because you heard from Paul, you heard from Jack and the others, there's the cell adhesion molecule. So this is the molecule which is really sticking out of the cell, making contact with the other cell, while we're saying is that probably this molecule, it's also regulating or participating in regulation of gene expression, which is quite a dramatic shift of understanding of the action of these molecules to what has been known. But we embarked on that. I don't want to say that it's 100% validated, that we are 100% happy with it, that it's happening, but we have fairly strong evidence that it's likely happening. We just need to kind of define how important is this, how relevant is this to the disorder and to the way how we can treat it. So we postulated, based on that PCDH19 most likely regulates or somehow co-regulates gene expression with estrogen receptor. Now, PCDH19, as I said, is an extracellular molecule. Estrogen receptor sometimes sits in a membrane and sometimes moves into the nucleus, where they regulates or co-regulates gene expression with many other molecules. There is a lot of evidence that estrogen receptor, especially recently, it's involved also in neuronal development. And there is also growing evidence that the mechanism of action is different in males and in females, which perhaps has not necessarily been taken into account in many studies, because mostly male animals are being studied, not many females, and very few investigators would study males and females at the same time. So there is also evidence that simply sort of activating this pathway just changes the way how the neuronal cells react. In this case, the estradiol increases neural outgrowth in these olfactory explant neurons. That's from not our work, so it's a published work of the others. So there's also good evidence that estrogen receptor is involved in neurogenesis, and you have seen these processes be involved in one way or the other in the presentations of other speakers today. So again, it's a signal comes from the outside through estrogen receptor alpha, estrogen receptor beta, or this other estrogen receptor, and that converges on different pathway into the nucleus, and then regulation of gene expression. So we set up this experimental system where sort of we either use the estradiol ligand or not. We use endogenous estrogen receptor and we deliver into the cell our reporter and our test. So we either put the PCDH19 or we put the PCDH19 with the mutation. We deliver this reporter, but we use the endogenous estrogen receptor as for the readout of the effect of protogalin 19. Now we do it in these breast cancer cell lines. I mentioned it last year in Rome. This is still the work which is under consideration in a journal. We're just revising the manuscript after very some useful comments from the reviewers. So we're using these breast cancer cell lines because they do express these receptors. So we just don't need to deliver another construct into specific cells. And unfortunately, these things moved a little bit. So we use these MCF7 cells, which express a lot of protogalin 19, and they express also estrogen receptor. And we use also these MDA-MB231 cells, which don't have as much PCDH19 and don't have estrogen receptor alpha. Now, we initially tested only a small number of mutations, these three, the frameshift and two missense. As you can see, there is a huge sort of emptiness in missense variants, so there are particular type of variation which have not yet been linked to PCDH19 epilepsy in this region. There are a lot of these protein truncating mutations which usually destroy the entire protein, so they are spread along the length. But these missense variants really cluster only in the extracellular region of PCDH19. So Yuyan has been doing this work now for a while, so she tested some of these mutations. So what we did, every time you see these different shades, so the darker one is with the ligand, with estradiol, the lighter one is without. So here we just do the vector, that's our background. Then we do PCDH19 wild type, and you see that there is a stronger readout, which means that the gene expression or the PCDH19 interacts with estrogen receptor and the luciferase activity of our reporter goes up. And when we put different mutations in, you see that the activity goes down at the level of the vector only or even below. And I have highlighted this one variant, just keep it in mind, that's one of the mutations. So this pretty much would mean to us that these variants in PCDH19 behave like complete loss of function. So we try to convince ourselves in many ways that this is the right readout, what we get. So we use actually the cell line here without the estrogen receptor alpha. And here you see that even the wild type PCDH19 doesn't do anything. So these are cells where there is a little bit of PCDH19, but the only component for the system to work, or the major component, which is missing estrogen receptor alpha. And then we don't see any readout. So that's how we believe that's another evidence that estrogen receptor is involved. But we're also doing knockdown studies to really further convince ourselves that we are on the right path. Now, we also found that PCDH19 using yeast to hybrid studies, and I'm not going to show you all the data, interacts with this protein which is called NONO. NONO is a paraspecial protein. It's a multitasker, and one of its tasks is to co-regulate gene expression with steroid hormone receptors and glucocorticoid receptors. So this is the link to corticosteroids. This is the link to progesterone receptor, estrogen receptor, and we found NONO to be interacting with the C-terminal part of PCDH19 using yeast to hybrid and co-immunoprecipitation studies. So when we tested again in the same system, PCDH19, and we put NONO as well, when you put NONO itself, you don't see really much difference with respect to the vector only, but if you put NONO and PCDH19, you see a stronger readout than if we just use PCDH19 only. So together these molecules most likely regulate estrogen receptor-mediated gene expression even more. Interestingly, if we use only the cytoplasmic region of PCDH19, we don't see this effect. Now, why we did it? We found that PCDH19 is cleaved. It's not the only protocoherent which is cleaved. So, as you have seen on some of the images of the previous speakers, this molecule sits in a membrane. But we found that sometimes the molecule is cleaved and the cytoplasmic region moved away. So we wanted to know whether that's perhaps not the mechanism how the gene regulation happens, but it's most likely not. It looks like almost the whole protocoherent goes into the nucleus and affects gene expression. And this is one of the reasons when we tested the cytoplasmic region with NONO, we didn't see the readout. And this is just on its own without NONO, so it's the same as pretty much the background. Now, NONO has been also recently found to be a very important neurodevelopmental gene, mutated in patients, with multiple patients with syndromic intellectual disability. NONO is also known to be involved in corticosteroid synthesis. And this is perhaps even the link to the methylprednisone, because that's a glucocorticoid, and that's exactly one of the receptors. And this molecule is involved in the metabolism of these. And another perhaps supporting evidence, but in view of course, Nicola's assessment, we see that Norimichi Higurashi, who was here a couple of years ago, they found that corticosteroids work well retrospectively on about 5 patients, and there's another paper about another patient where corticosteroids achieved sort of amelioration of seizure activity. Perhaps would fit, but this can be also biased because these patients might be just having a gap between their clusters. So that's to be assessed, but if you look at it kind of positively, it kind of would make sense because this PCDH19 NONO estrogen receptor axis is likely involved in metabolism of these substances. So when we found this link, and we're still investigating in greater detail, we also thought that it might be a way how we can sort of separate the wheat from the chaff. That's how we can say that which PCDH19 variant is relevant for that patient, or the family needs to look for another cause or another explanation of the disorder. So it is important because this information gives us advice which variants are disease driving, and that has huge importance for clinical trials, but also for the understanding of the mechanism of the disease. As I showed you, there are the pedigrees where the women seems to be less severely affected. There are the singletons where these girls seem to be more severely affected. So there was still a possibility that maybe there are different types of variants in these girls, perhaps acting through different mechanisms, which is the case for several other genes I've been working with. It looks like that all the variants are just loss of function, so it's probably the explanation is not in a type of variant, but we formally had to investigate that. And one thing which I would like to really stress, I'm stressing it at least twice, that even though we see the functional effect, it doesn't mean that disease variant is really disease causing. And that's a difficult question. It's really a hard task that we see that the neuron looks different and the neuron moves differently. It doesn't mean that that's the cause of the disorder. 20% of all nucleotide variation, missense variation, even common variation is predicted to have functional effect. So we're going to see changes of function, not necessarily the change of function explains what we see in a patient. Now, how we can get around that difficult task is not obvious. We just need to collect multiple lines of evidence, clinical, molecular, to satisfy that answer that is or it's not a disease driver. So we tested different types of variants. was all more complicated because we could not test these variants in a fraction of the molecules, so we had to make the full-length constructs. And this was also difficult because when we use different techniques in the laboratory, these molecules accumulate a lot of variations, so we actually decided to synthesize all these 20 plus variants. Then we need to validate them, and we need to check for expression. So there are a lot of work behind which I'm really not showing you to assess these initial 20 plus variants. But generally we had 4 categories, or 3 main. Well, we tested these 3 mutations which are in red, then we selected quite a few other mutations from other published cases with PCDH19 where we believe that they ought to be disease driving. Then we selected a few variants where we see some low population frequency, like there are 4 or 5 alleles in some of the databases, like the Exome Annotation Consortium, for those who know what I refer to. It's a collection of about 60,000 individuals where we can look how frequent these variants are, because most of these variants otherwise are fairly unique so far, so they're not found in anybody else. And then we also collected a few common variants, so variants which are probably found in 1 in 1,000 individuals or 1 in 100 individuals, which technically shouldn't do anything, so they were serving as controls for our experiments. So we have done all these tests, and these are, for example, the mutations. And as you can see, this is again the vector with the stimulation using estradiol. The estradiol is always as a control for us because we always should see a bump of extra activity after put the ligand, because the ligand will interact further with the receptor and increases its activity on gene expression. So here the mutations all look pretty much like almost loss of function with respect to the wild type PCDH19. Then we tested these variants of unknown significance. Now here you can see it starts looking a little bit more complicated. This is the vector only, this is the wild type, and there is kind of a range. Well, and then you can say, well, is this, for example, different to the wild type or is not? So this is really also explains to you the difficulty for calling these variants of unknown significance, which might be just not necessarily disease driving, but they might be contributing. Depends on which model you adopt, whether there is a threshold model that you need this much, that much, that's its continuum of activity, or there is activity and there is no activity. But we can classify this when we use additional criteria, not just this particular expression. I'm going to talk about it. And then there are a few common variants here, which pretty much look like wild type. And that was good to see because these frequent variants, based on their frequency in a population, we can just say that they are unlikely to be causing any problems, and that's pretty much what we see. They behave like any other protogenerin 19. Now, when we were doing our gene expression profiling, we found that several downstream genes were deregulated, and I'm showing here oxytocin receptor itself, and the mouse model, our model of autism spectrum disorder, and you probably would have heard about clinical trials of oxytocin in autism, and that's why deregulation of oxytocin receptor is relevant to the behavioral phenotype. ENC1 is a known transcription factor which is involved in cell fate determination. Again, it's been changed, and apolipoprotein D, it's an important cholesterol transporter, so it's a molecule which is very important for the synthesis of the steroid hormones, and that has been deregulated. So in this case, on top of these studies, what we do, we go to these cells when we stimulate or not with estradiol, and when we transfect or modify the cells with the wild type protocoherin or the protocoherin with a variant of of unknown significance, which we need to test. Then we collect the cells and we also look for the gene expression. So we don't look at our reporter, which is the luciferase, which is a bright protein which can easily and very sensitively measure. But we also look at the genes in the cell and how they respond, because they should be the targets of this specific transcription factor. And as you can see, this is just a vector only. This is when we put the wild type, and these gray bars are when we put the mutant. So in this case, you see that the mutants are behaving very differently. And we want to see both. We want to see the wild type, sometimes increasing, sometimes decreasing. The gene expression depends how they act together with estrogen receptor. And we want to see also that once we put the mutant in, we pretty much lose these functions. So that's kind of another validation step for us to make sure that the system is working and that we're getting a good readout. And we have a good 20 of these genes which we can measure. Among them are also glutamate receptors and other interesting genes like the neurosteroid metabolizing enzyme, the ACAR1C. So, and they all seem to be behaving as we expect, but the experiments have not been completed for every single mutation to look at every single one of these genes yet. Now the other tool is, of course, looking structurally at the protein, the way how the protein folds, because the folding of the protein can predict its function. And coherent repeats, especially the region which is sticking out. You saw PCDH19 has 6 of these. The structure is known, so we can model the effect of these mutations on the structure of the protein, and based on that predict how it's likely going to act. In this case, this is the mutation which I highlighted very quickly, which behaved in our tests as complete loss of function. With the estrogen receptor reporter assay. Here, the blue structure here on the background, and here a little bit, that's one of those extracellular coherent repeats. And if you just change this one little from serine to proline, the organization changes from the nice blue one into this red one, which you look, this completely would disjoin the structure. It looks different, and as you remember Paul's pictures how these protocoherin interacts, that would pretty much destroy the ability of this protocoherin to interact with a similar protocoherin on the other cell. So it would render it pretty much this loss of function effect. The other experiments, what we do, as Jack mentioned, we use these different mixing experiments. In this case, we used the neuronal stem cells from the knockout mice Paul generated. We have the wild type mix and the knockout, and we took them into differentiated neuronal stem cells, and we had done gene expression profiling. I don't have very much time to go into detail, but what was really interesting, the most significant pathway which popped up was in estrogen receptor signaling. Already at that stage, they are slightly different genes. They are not those genes which I showed you, like apoprotein D. Probably those genes are perhaps irrelevant at that very, very early stage. But the most significant pathway which came up was estrogen receptor signaling. And another work, what we have and perhaps can be used to test the PCDH19 variation are these neurospheres assay. This actually experiment is part of the knockout work Paul, mouse knockout work Paul has been driving, where we found that when we looked at the wild type versus knockout telencephalic neurosphere, so these are bundles of kind of stem cells from which the neurons, the stem cells sort of differentiate into neurons, and the neurons migrate out into in vivo in their destination, but on a dish they just migrate away. And if you look how far they go, you see that PCDH19 neurons kind of go a little bit further in certain areas. They kind of either they go earlier, they start earlier, but they go further. So there is this neuronal migrational phenotype, which to some extent perhaps may reflect what other people are finding even in zebrafish when they were using morpholino technology. Also the final phenotype was a lot more severe. So again, it kind of makes sense that there is a neuronal migrational phenotype. Nobody had mentioned it today, but PCDH19 was found to be part of this wave complex, which is important acting inside the skeleton remodeling complex, which actually does make perfect sense for these molecules to have effect on dendrogenesis, neurogenesis, and neuronal migration. Now, this is just a couple of concluding slides just to really say that the assessments of this variation is very important. Many people really focused on it. We can do it many different ways. We can do it using computer. We can go into the laboratory and to gather additional evidence. For PCDH19, it's really the challenge is that we see many of these individuals not necessarily being affected. We have that problem of non-penetrance, and plus we have the males who generally do not present with the typical disease. Our colleague now recently deceased, Dick Cotton, has written very interesting articles, have been instrumentally driving an international project on human genome variation. And he very well predicted a long time ago that the missense mutations are going to be the hardest because they are just very subtle in their appearance, but they may have really dramatic consequences. Other people, like our colleague from Australia, Dan McCarter, who is now in Boston, were really trying to spur a lot of activity to offer some guidelines, and really the list of these is going on and on and on. So this is a challenge for us as scientists, this is a challenge for the diagnostic laboratories, and it's something of huge importance to get right, because when it comes to the clinical trials, when it comes to feed this information back to the families with these individuals, we really want to get it right. It is not how it used to be 10, 20 years ago, that they just because the mutation happens de novo, it's disease causing. I mean, we were as blunt as that. As soon as we saw it de novo, that mum didn't have it and dad didn't have it, we really jumped very quickly to the conclusion, well, this must be disease causing. Now we know that each one of us have probably about 2 de novo mutations, which our parents don't have, and likely happen in the sperm or the egg, or very early during our development. So it is becoming really a challenge, and because with that comes the question of interpretation of these variants, correct diagnosis, and as well as prognosis, because that's where the value of genetics is, that we don't have just a diagnosis, we're also going to have prognosis. And really the importance for personalized medicine, which we heard today multiple times. But again, I would like to stress that not all functional variation is disease relevant. And that's really a task which really requires the combination of all of these important dot points and the evidence to conclude. So if I was going to conclude, I have 2 summaries. So we believe that really the testing of these variants is not easy, but it's really very important. And I put here the question mark that how much do we trust our traditional gene manipulation techniques? Now I refer here also to the work Paul referred in his paper, where the downregulation of PCDH19 in zebrafish really generated very severe phenotype. Really the neuronal migrational disorder with obvious brain malformations, which he didn't see in his mice and we don't see in the patients. So really the question is what's going on, how much these acute gene manipulation technologies are telling us about the phenotype. Fortunately, the human cell has really the huge ability to compensate for a loss of gene function, especially when the gene function is lost right at the beginning. And that's why generally these phenotypes are not as severe as the acute gene manipulation phenotypes, and we need to be really careful how we interpret that data. Not all variants affect gene function. That's a reiteration of what I said. We really haven't ventured into non-coding variation. And we do it for some of the disorders, but I haven't seen really any mutations as yet in PCDH19, which would be regulatory, like duplications or promoter variants. Not many people actually looking whether that would affect expression of that gene. And generally, using a single approach is it's not necessarily the best way forward, especially when you come across a unknown variant. And we believe that really these individual genes will need to generate toolboxes. That's what we try to generate for PCDH19 to really be able to interpret this variation correctly. In a big picture, we see that we're finding more and more in these individuals. We're also finding individuals are perhaps wrongly diagnosed simply because the diagnostic laboratories are not necessarily capable of predicting the effect of this variation. We found that PCDH19 interacts with this paraspeckle multitasker, Nono, who is also involved in circadian rhythm control, and whether this is relevant to the rhythmicity of the seizures, well, is yet to be determined, but it's certainly an important regulator of glucocorticoid and corticosteroids, and these have been found to be efficient at least some patients with PCDH19 epilepsy. And that's captured here to some extent. Now, we still struggle to really understand and see, even from post studies, that PCDH19 go to the nucleus and it's enough of it there, that it does regulate, that it's actually the whole this pathway is relevant. But our data so far shows that this is likely happening. It's just, we need to figure out when it is actually crucial and what slice of the problem it takes, because clearly PCDH19 is not going to be only due to the deregulation. Of course, you see it's a migrational problem, it's a developmental problem. But what we're looking at is really the terminal situation where we see the neurosteroid hormone deregulation in the blood of these individuals, which perhaps is telling us where the system stops and what the readout is, metabolic readout of the system, and that's what we try to fix. It's also telling us about the broader importance of estrogen receptor. And I didn't talk very much about progesterone and androgen receptor, but I suspect from our first studies that these receptors can be regulated by PCDH19 as well. And that's perhaps to some extent may complicate the whole story even more. But the practical outcome is that now we have another test together with existing tests which we can use to really determine if we need the effect of this variation, at least along this pathway, if we believe that that is correct. But it provides us another tool. Really, for this specific work, there are a lot of other people we work with, but for this specific work, it's the people in my laboratory and in Paul's laboratory who contributed the most. And I really appreciate the support of the Italian and the American PCDH19 parents and their friends and colleagues to advance this work. So thank you for your attention.
Questions for Jozef? I actually have one question. So in your summary or your conclusion slide, acknowledging that we don't know if we trust our own models. So as a clinician, hopefully Nicola will back me on this. So what if the phenotype fits perfectly and you get a variant of unknown significance and yes, it's de novo? Why should I trust anyone who's spoken so far to say that no, it doesn't perturb any of our models, it can't be the cause of the disease? How do we reconcile that? I mean, this is where, for me, it seems if the phenotype is classic and there is a mutation, then I still rely on that. So convince me otherwise.
Yeah, I said you are a busy clinician. You have probably 10 minutes, so you act instinctively. There's many examples of exactly the same phenotype or apparently the same phenotype caused by different mutations. So we actually tend to simplify matters as soon as we can because it gives us an answer. So our brain probably channels us into this answer. Yep, this has 2-3 characteristics of what I'm used to, and plus they found a variation in PCDH19. Doesn't matter where it is or what it does, but it does make sense. So tick. I guess that's, I mean, the extreme of the explanation. I don't know whether you're convinced. I wouldn't be convinced after the answer like that, but that's perhaps sometimes we see it. That we want to have that case resolved, and all the evidence points towards that. But we have to realize that in terms of genetic variation, we are not that good in predicting its effect, and actually in opposite. Many cases we have to go back. For example, in intellectual disability field, we had to withdraw 10, not we, but as a field, we probably went back, and 10 genes were almost taken out of the list, because the evidence was not good enough over time to support their involvement. And again, all the evidence initially looked obvious, but as the time told us, it was not. So, yeah, we need to be a little bit more careful. I agree with you if the phenotype fits. It's a very strong argument that you are on the right track, but the assessment of the variation probably need to be a little bit more thorough, provided that we have tools for it, because clearly not many genes, actually I would say a small minority of genes would have tools so we can do it experimentally.
So maybe a follow-up to that, if the phenotype fits perfectly, yet the models don't predict it as being disease-causing, is that actually helpful to you to figure out, are there some other ways in which PCDH19 could be interacting with yet to be determined molecules or pathways?
If you're alluding to really other modifier genes or other modifier mutations and multiple clinically relevant DNA variation findings, we see more and more, and we already discussed it to some extent. Richard Gibbs told us from their 15,000 clinical exomes that as soon as they see a phenotype which is a little bit more variable, they actually have they are more likely to find more than one clinically relevant variant. Up until now, we have stopped looking for other variants as soon as we find one, because we already have something. But we know, and there are people in the room and there are people outside the room, where we studied 2, 3, 4 clinically relevant variants in these children. And that's really a very hard task because they generally fit the phenotype for all 4 of them, or at least 3 of them. But we don't have the conclusive evidence that which one is actually driving it, if there is one.
Well, my daughter, she does have the D121H disease-causing mutation, and she fits the whole bill for everything. Where her, I have her report right here, and it says that it's associated with epilepsy, febrile seizures, intractable seizures, growth delay, poor weight gain, easy fatigability, constipation, kidney stones, leg pain, recurrent infections, recurrent viral infections, herpes simplex, encephalitis, chronic otitis media, allergic rhinitis, and reduced natural killer cells, and immunodeficiency. And at 13 months when she started having seizures, it was a febrile, it was GI bug, and we found out later that it was, she had C. diff, and then at 15 months she had a cold sore, and a cold sore that we treated with acyclovir, but it went into her bloodstream. And of course, at that point, she was on phenobarbital and dilantin, and they switched her over to Keppra. And so after we figured out the allergic reaction type parts to that, we had some control, 2 years seizure free, and then at age 9, things went nutty. And but when I went back, hindsight, we always went to the hospital. They always did a PCR viral panel to check to see what she was fighting, because I was always just gut intuition. They had originally diagnosed us with complex atypical febrile seizures, and so we had that for 8.5 years until we officially got the PCDH19 diagnosis in August of 2014. But she would be positive for flu A, flu B, rhinoenterovirus, these common things that all doctors shut us down, because I'm like, she seizes on swine flu, she seizes on immunizations, especially the live viruses. And so, as a mom seeing all this, and then of course we didn't realize that it was going to pay off later on, that we actually have documentations of her every time she went in. Now it's not so much because we try to keep her out of the hospital because it seems to make it worse when we go in. But the kid has no immune system right now. And of course she just had to leave because she had a seizure just a moment ago. But the bottom line is she fits her mutation. And, for us, we go to our doctors, and we have a doctor in Miami. She's very open-minded, and her partner not so much. And it just depends on who you go to. And, as parents that are fighting this fight, we're having to stand up to doctors that are not really open-minded to this stuff. And a lot of them do not know a lot, and we know more than they. And so it just becomes this big old ego battle, and you're just sitting there fighting for your kids' lives, And when we have now, I always told them she's seizing on all these viruses. And what the crazy part is Recefin, which is a bacteria coverage, gets her over the hump. And we can't explain it. But, I started going into functional medicine just to try to branch out and see where we needed to go. And, of course, they're all into intestinal permeability, which my Eastern doctors call it leaky gut. Western doctors call it intestinal permeability, and they're saying that it's the gut and brain connection. And so, you look back, and this journey has been going on for a while, and she fits everything, but it's just, I know everyone wants scientific proof, but there's just sometimes you just don't get that scientific proof, and even when you get it, they still want to argue it. And so I don't know. She definitely fits her bill of her mutation. We've had the whole exome gene sequencing, and this is the only thing that she has, and it's de novo. We've all three been tested, my 14-year-old and my husband and I. So we're definitely one of the families that fit the bill for her specific mutation.
Yeah, you very nicely, if I can comment, and I can others to comment to it, you described the difficulty sometimes to link really the problem to perhaps genetic or non-genetic cause. And please don't be too hard on the colleagues when they try to find the answer for you, because if you look at it over my time, the lifetime, we started really from 2 or 3 or maybe 5 genes we knew cause inherited disorders to about 2,500. At the moment, we have about 800 genes causing learning disabilities. We have about 300 genes causing epilepsies. We have about 800 genes causing autisms. And really for specialists, even for basic scientists, it's really very hard to keep up and to be on top of everything. I guess what we do need to learn is just also listen more to the parents, because parents usually do a lot of work. They know the children much more. But it's really a fine balance. In terms of other sort of reactions and problems which can sometimes complicate the diagnosis, we all have other variation. We can be more sensitive to specific drugs, and even the drug is working well on the other patients, but we may have a genetic variation which prevents that drug to be used in your specific case, simply because we have that genetic variation which makes it complicated. And that really, those patients don't necessarily fit the bill, and that's why the specialists are trying to find very hard the answers, and that's why there are probably differences of opinion. I don't know what is the best solution for it. The best is really just to keep asking, putting brains together, and organize a meeting like this where we really exchange the information and educate each other, because really we can't know everything ourselves. We really need to take advantage of the common and brain when we're tackling these very, very difficult questions. Joe, would you like to comment on the
No,
diagnostic odyssey?
I hope I was not coming across as confrontational with Joseph, and neither he back. I think it was just trying to understand, despite all of the knowledge that we now have, we are realizing we know even less, and it gets very, very complex. And so I
You
think
are the first participant
one of the
to join the
purposes of
conference.
symposiums like this is to bring together the clinicians, the basic science researchers, and the parents so that we can all get to where we want to be.